If you will recall, cell theory states that all life is composed of cells, and all cells come from pre-existing cells. So let's investigate the smallest unit of life in more detail. So where are the cells? If I told you there was a picture of a cell on this page, would you believe me? If the picture were the actual size of a cell, there could be. Because the vast majority of cells are microscopic, meaning they cannot be seen by the naked eye. We measure cells in micrometers. If you look at this scale, a micrometer is smaller than a millimeter, but larger than a nanometer. There are 1,000 millimeters in a meter. But there are 1,000 micrometers in a millimeter. That is an extremely small measurement, and our eyes cannot make out objects at the micrometer level. Many structures we find in cells are even smaller and can be measured in nanometers. There are 1,000 nanometers in a micrometer. So to investigate cells properly, we must use microscopes. This is a compound light microscope, similar to the one you use in lab. A light microscope transmits light through a specimen and magnifies the image with a series of lenses. A light microscope can magnify a cell about 1,000 times its normal size. We can visualize even smaller structures or examine cells in different ways with electron microscopes. Electron microscopes use electrons instead of light to produce a magnified image. We can easily visualize structures inside cells with a transmission electron microscope, or TEM. The mitochondrion is found in eukaryotic cells and is about a micrometer across, but we can even see the nanometer-sized structures within the mitochondrion. A scanning electron microscope, or SEM, is not typically used to visualize the interior of a cell or other structures. Instead, it produces a 3D image so that we can see what the surface of a specimen looks like. Now that you know how scientists study cells, we can examine how cells are organized. While there are two basic cell types, prokaryotic and eukaryotic, all cells share a few features. All cells are surrounded by a plasma membrane. All cells contain an aqueous solution called cytosol, or cytoplasmic fluid. All cells have DNA as their genetic material, which is organized into structures called chromosomes. And all cells have ribosomes to synthesize proteins. The prokaryotic cell is relatively simple. Prokaryotic cells may contain a few organelles, but most do not possess them. Prokaryotic cells also lack a nucleus. Organisms composed of prokaryotic cells are called prokaryotes, and can be found in the domains archaea and bacteria. Prokaryotic cells are relatively small, about 1/10 the diameter of a eukaryotic cell. The bulk of the genetic material is organized into a circular chromosome and is found in a region called the nucleoid. The nucleoid is not surrounded by a plasma membrane. A few genes are not located in the nucleoid, but can be found scattered throughout the cell. These small DNA molecules are called plasmids. Granules of starch and other carbohydrates may be stored in the cytosol for later use. A cell wall containing peptidoglycan is found exterior to the plasma membrane. Extensions on the cell surface called pili are used to transfer plasmids from one bacteria to another. Every other organism is composed of eukaryotic cells. These cells are characterized by highly organized membrane-enclosed organelles and a prominent nucleus which contains the hereditary material DNA. Organisms composed of eukaryotic cells, animals, plants, fungi, and protists, are called eukaryotes. Here's a generalized animal cell. The contents of a cell are referred to as the cytoplasm. Within the cytoplasm, we find the organelles that compartmentalize specific chemical reactions. The plant cell has the same basic components as animal cells, but contains chloroplasts and other plastids, cell walls, and central vacuoles. Plant cells do not contain centrioles. As you learned from the last lecture, dynamic membranes surround cells. These lipid bilayers also surround organelles. Membranes isolate the contents of the cell from the external environment, regulate what substances move in and out of the cell, and allow cells to interact with different molecules and other cells. Since the phospholipid tails in the membrane are hydrophobic, a selective barrier is formed between the cell's contents and its environment. Channel and carrier proteins embedded in the membrane allow necessary substances to move in and out of the cell. Glycoproteins facing the extracellular fluid communicate information to other cells, while proteins facing the cytosol may bind to and stabilize filaments in the cell. The centrally located nucleus is the most prominent organelle. Here's the nucleus. It contains most of the genes in the eukaryotic cell. It averages 5 micrometers in diameter. The nuclear membrane is surrounded by a double membrane called the nuclear envelope, which is perforated by pores. The DNA inside the nucleus is packaged with proteins and referred to as chromatin. Also inside the nucleus is the nucleolus, the site of ribosome synthesis. This is a diagram of the nucleus. The nuclear envelope separates the nuclear contents from the surrounding cytoplasm. At intervals, the double membrane comes together to form nuclear pores, which regulate the passage of materials into and out of the nucleus. This micrograph of the nucleus has been colored to show the nuclear pores in pink. What kind of microscope was used to produce this image? Yes, an SEM. Within the nucleus we find the DNA, which is packaged with proteins into chromatin. On this micrograph, the chromatin is visible as a grainy substance. What kind of microscope was used to produce this image? Yes, a TEM. Ribosomal RNA, or rRNA, is synthesized in the nucleolus. In addition, proteins are imported from the cytoplasm into the nucleus. Within the nucleolus, these proteins and RNA molecules are combined to make the small and large subunits of the ribosome. The subunits leave through the nuclear pores and are assembled in the cytoplasm into a ribosome. This micrograph shows the nucleolus as a dark structure within the nucleus. So what are those ribosomes made by the nucleus? Ribosomes are the site of protein synthesis, as they put together amino acids based on instructions found within the DNA. So the information on how to build proteins is found in the DNA. An RNA molecule transfers those instructions to the ribosome in the cytosol, and the protein is made. We will discuss protein synthesis in more detail in our genetics unit. Again, ribosomes consist of a large and small subunit that are composed of proteins and ribosomal RNA. Free ribosomes can be found in the cytosol, or bound to an organelle called the endoplasmic reticulum. The endoplasmic reticulum, or ER, is attached to the nucleus. Here's the ER. It is a complex of membranes that encircles the nucleus and is continuous with it. The pouches formed by the folded membranes are called cisternae, and the internal space of the ER is called the lumen. The ER membrane and the lumen contain enzymes that catalyze reactions. There are two distinct but connected regions of the ER that differ in structure and function, the smooth ER and the rough ER. Rough ER is studded with ribosomes and plays a central role in the synthesis of proteins. Polypeptides are produced by the bound ribosomes and transported into the ER lumen. Smooth ER is tube-like and synthesizes lipids. Enzymes within the lumen catalyze reactions to produce phospholipids, cholesterol, and hormones. It also detoxifies certain poisons and stores calcium ions. This micrograph shows a false color image with the cisternae of the ER yellow and the ribosomes red. This is an SEM image of smooth ER found in a liver cell. An abundance of smooth ER is found in liver cells as enzymes in the smooth ER break down alcohol and drugs. This diagram shows how both the membranes of the rough and smooth ER are continuous with the nuclear membrane. Products produced by the ER are contained in bits of membrane that butt off the ER called transport vesicles. The vesicles and their contents typically go to the Golgi next. The Golgi is the processing center for ER products. Like the ER, the Golgi consists of cisternae, and its internal space is also called a lumen. However, the Golgi differs in function. The side closest to the ER, the cis face, receives the transport vesicles from the ER. In the lumen, enzymes modify ER products, which are then repackaged into another vesicle. A molecular label is also added to route the modified product to the correct location. The repackaged product buds off the trans face of the Golgi. This diagram shows the passage of an ER product through the Golgi. Try to find the cis face of the Golgi in this micrograph. Yes, it's the top. Lysosomes are produced by the ER and the Golgi. Lysosomes are not easily distinguished from other vesicles, but this could be one. Lysosomes contain digestive enzymes that hydrolyze macromolecules. The enzymes are proteins produced in the rough ER, which are modified and packaged in the Golgi for export to the cell interior. There are several different substances that may need to be digested by cells. Some cells ingest other organisms. For example, an amoeba may engulf a bacteria through phagocytosis. The cell membrane would surround the bacteria to form a phagosome. A lysosome would then merge with the phagosome and digest the bacteria's macromolecules. The resulting monomers would be used to synthesize necessary cell products. In autophagy, damaged or unnecessary structures would be marked for destruction by lysosomes. For example, this mitochondrion is worn out. A membrane would surround the mitochondrion. Then a lysosome would merge with it, and its hydrolytic enzymes would digest the organelle. The resulting monomers would be recycled in the cell. It is a storage organelle for proteins, noxious substances to deter herbivores, water, food, salts, pigments, and waste. Central vacuoles also contain hydrolytic enzymes to break down waste and other cell components. The vacuole is hypertonic to the rest of the cell, and this osmotic pressure allows plant cells to be turgid. 50 to 80% of the cell may be taken up by the central vacuole. Mitochondria are the site of aerobic respiration, an oxygen-requiring process that converts chemical energy to ATP. Mitochondria are found in all eukaryotic cells. Here are the mitochondria. A single one is called a mitochondrion. Mitochondria are easy to spot. Each mitochondrion is enclosed in a double membrane. The intermembrane space lies between the outer and inner membranes. The inner membrane folds inward, producing sac-like structures called cristae. The matrix is the area within the inner membrane. This diagram shows the locations of the outer and inner membranes, the intermembrane compartment, and the sac-like cristae. Make sure you can locate those structures on this micrograph of a mitochondrion. The chloroplast is the site of photosynthesis and can be found in plant and some protist cells. Here are the chloroplasts. The chloroplast also has a double membrane. A space called the stroma is found within the inner membrane. Found in the stroma are thylakoids. Thylakoids are membrane pouches that contain chlorophyll, the molecule that captures light energy. The thylakoids are stacked on top of each other to form grana. This diagram shows the locations of the outer and inner membranes, the stroma space, and the thylakoids. And there are plastids other than chloroplasts in the plant cells. These cells are from a potato and contain amyloplasts, which store starch. The cells have been stained with iodine so that the starch can be better visualized. It is hypothesized that mitochondria and chloroplast originated by endosymbiosis, in which a larger cell engulfed, but did not digest, a smaller cell in the past. The evidence? Well, both organelles are similar to bacteria in structure and have their own circular chromosomes. Peroxisomes are small organelles that contain enzymes that oxidize fatty acids and amino acids, as well as detoxifying some poisons, such as alcohol. A byproduct of many of the reactions that occur in peroxisomes is hydrogen peroxide, which can damage membranes. Therefore, the organelle also contains an enzyme called catalase, which decomposes hydrogen peroxide to water and oxygen.